the spine of zebrafish, which plays a positive role in disc maintenance and potentially regenerating aged disc in vertebrae, may hold promise for medical interventions. In humans, spinal discs naturally weaken over time, leading to various health issues including lower back, neck, and appendage pain. Currently, there are only symptomatic treatments available for weakened disc in humans, which involve pain relievers or anti-inflammatory medications. In more severe cases, disc replacement or disc fusion surgery is performed. Therefore, there is an immediate need to develop a treatment process based on enhancing the function of weakened disc or regenerating disc in humans. A study conducted by the Department of Science and Technology (DST), Government of India at the Agharkar Research Institute (ARI) in Pune has identified a protein called cellular communication network factor (CCN2A) secreted from intervertebral disc cells that inspires the regeneration of weakened and aged disc in zebrafish spines. It does so by modifying the FGFR1 SHH fibroblast growth factor receptor sonic hedgehog pathway, enhancing cell proliferation and protecting cells. This study, utilizing zebrafish as a model organism, represents the first in vivo study demonstrating the potential to activate an endogenous signaling cascade to inspire disc regeneration in weakened and aged disc. Scientists have also found that CCN2A FGFR1 SHH signaling cascade plays a positive role in both disc maintenance and the process of disc regeneration. Published in the journal Development, this study employs genetic and biochemical approaches, shedding light on the genetic and biochemical aspects of the process of weakened disc function and disc regeneration. It could pave the way for developing a new strategy to slow down the degeneration process or inspire disc regeneration in weakened human disc. For more details, click on our website www.aajkiawaz.in.